Hello students, we welcome to EPG Pathshala. I am Dr. Asutosh Rai from Indian Institute of Vegetable Research, Varanasi. Today we are going to talk on module Plasmid, Lambda-based vectors and derivatives under the paper Genetic Engineering and Recombination DNA Technology. After completing this module, you have to able to understand plasmid, that is vehicles for cloning, lambda bacteriophages and derivatives of lambda bacteriophages. Plasmids. Plasmids are the extrachromosomal self-replicating DNA found in bacteria. The size of plasmid DNA range from 1 kb to 250 kilobases. Almost all plasmids are double-stranded circular DNA, conferring an extra phenotypic character to the bacteria. Closed circular DNA of plasmid vector are cross-coiled over its own axis in three-dimensional space to form a supercoil. These characters are may include antibiotics resistance like ampicillin, tetracycline, or kanamycin resistance, etc. Abiotic stress tolerance like heat, cold, salt, or toxic species resistance, or may responsible for synthesis of some special polypeptides like toxins or metabolites. Plasmids can be classified as plasmid can be classified on the basis of various features found in it. We have categorized few of them like fertility or F plasmids having a tragene which enables bacterium to conjugate through X pili. Resistant plasmids, these are R plasmids containing antibiotics or poison resistant genes conferring antibiotic resistance to the bacterium, also known as R factor. Col plasmids are genes that transcribe and translate for bacteriocin. These proteins are responsible for killing of other bacteria. Degradative plasmids provide the capability to bacterium for degradation of various organic or inorganic substances which are harmful for bacteria. Examples are toluene or salicylic acid resistant bacteria. Another group is virulence plasmids. These virulence plasmids confirm pathogenicity to the bacterium. Naturally occurring plasmids have several limitations. For example, some stringent and non-relaxed plasmids. These are the example is plasmid SC101. It carries only 1 to 2 plasmid per host cell. Some has poor marker genes like COL-E1, and some are too large, can be handled. To overcome the limitations of natural vectors, artificial plasmids are designed for combining different elements from diverse sources. Artificial plasmid vectors are classified into two broad types based on their use. First is cloning vector or expression vector. Apart from the following, there is another class of vector known as subtle vectors. Subtle vectors can propagate in two or more different host species, both in prokaryote or eukaryote. Hence, inserted DNA can be manipulated and replicated in two different cellular systems. Cloning vectors are designed for efficient transfer of foreign DNA into the host. Expression vectors have efficient machinery for cloning and expression of foreign genes in the host system. Selection of a vector depends upon the various criteria decided by the experimental goal. A good cloning vector should have the following characteristics. It should have autonomously replicating. It means that it should have a region or origin of replication. A vector should contain at least one selectable marker like gene of antibiotic resistance and It may contain a reporter marker like beta-galactosidase or green fluorescent gene, etc. Presence of unique restriction enzyme site. This region is also known as multiple cloning site or MCS, preferably small in size and easy to handle. Relaxed control of replication to obtain in multiple copies. Presence of appropriate regulatory elements for expression of foreign genes. The selection of suitable vector system depends mainly on the size limit of insert DNA and the type of host intended for cloning or expression of foreign DNA. Origin of replication. The initiation of DNA replication does not occur at random sites around any genome, but rather it is initiated at a specific point called as origin of replication. Once the DNA synthesis has been initiated, two replication forks extending in either direction from origin of replication proceed to allow the full replication of the genome. Bacteria such as E. coli have a single origin of replication called ORIC. Eukaryotic cells, on the other hand, have multiple origins of replications which are different from ORIC. The yeast Saccharomyces cerevisiae have been estimated to have about 300 replication origins, whilst human cells utilize over more than 20,000 origins of replications during replication of its genome. ORIC is a 245 base pair DNA sequence that acts as the binding site for a number of proteins, namely DNA A, DNA B, and DNA C protein. The binding of these proteins promotes the melting or opening of DNA helix, a process that is essential so that the DNA replicating enzymes can read the base sequences. Replication origins in both prokaryotes and eukaryotes probably serve the same overall function, but the replication origins of prokaryotes will not substitute for their eukaryote counterparts. Plasmid vectors have multiple cloning sites. Multiple cloning site a short DNA sequence consisting of restriction sites for many different restriction endonucleases. Multiple cloning sites escalate the number of potential cloning strategies available by extending the range of enzymes that can be used to generate a restriction fragment suitable for cloning. By combining them within a multiple cloning site, a site are made contiguous so that any two sites within it can be cleaved simultaneously without exceeding the vector sequence. The multiple cloning site is inserted into the LAC-Z sequence, which encodes the promoter and the alpha peptide of beta-galactosidase. Insertion of the MCS into the LAC-Z fragment does not affect the ability of the alpha peptide to mediate the complementation, while cloning DNA fragment into the MCS does. Therefore, recombinants can be detected by blue-white screening on the growth medium containing X-gall in the presence of IPTG as inducer. The selection of transformed cells from the non-transformed population is done by using selectable marker gene that may confer the antibiotic resistance. Hence, the cells only having the vector with the resistance gene for the antibiotic would grow in selection medium containing the antibiotic like ampicillin, tetracycline, etc. A white and non-transformed cells would die. Here, some antibiotic and their respective genes present in plasmid are being presented. Selection of transformed cells from non-transformed population depends on antibiotics. You can see here the antibiotics ampicillin, kanamycin, neomycin, chloramphenicol, and gentamicin, tetracycline, and streptomycin genes. Copy number means the number of plasmids present per cell. On the basis of copy number, a plasmid is called relaxed plasmid or stringent plasmid. A relaxed plasmid are maintained in at multiple copies per cell, like 100 to 200 plasmids can be found inside a single cell, while the stringent plasmids are present at a single copy or a very low number of copies of plasmid in between 1 or 2 per cell. This table represents the common reported gene markers which are used in day-to-day cloning activities. Here you can see the genes, their proteins, and the size of amino acid sequence found in that protein and their source of origin. You can see here CAT gene, which is responsible for chloramphenicol acetyltransferase, LACZ for beta-galactosidase, GUS-A, LUC, that is luciferase gene, and green fluorescent protein gene, which was isolated from jellyfish. PBR 322 is a widely used E. coli cloning vector, that is plasmid vector. It was created in 1977 in the laboratory of Herbert Boyer at the University of California, San Francisco. The P stands for plasmid and BR for Bolivar and Rodriguez, researchers who constructed it. You can see here the various restriction sites, then origin of replication, and resistant gene for ampicillin. It have the origins ORIC in its structure. PBR 322 plasmid vector have the following elements. It have a rep gene, that is replicon from the plasmid PMB1, which is responsible for replication of plasmid. Second, it have a rope gene that encode rope protein. Rope protein are associated with stability of RNA1 and RNA2 complex and also decreases the copy number. The source of rope gene in PMB1 plasmid, the tetracycline gene, that is TET gene encoding tetracycline resistance, derived from plasmid SC101 plasmid. A BLA gene encoding beta-lactamase, which provide ampicillin resistance. Its source was transposon TN3. PUC18 plasmids are small, high copy number plasmids of size 2686 base pairs. This series of cloning vectors are developed by Messing and co-workers in University of California. The P in its name stands the plasmid and UC represents the University of California. There in diagram, one can see the various restriction sites in MCS multiple cloning site region of the antibiotic resistance gene ampicillin lac-operon region can be seen. PUC vectors contain a LACZ sequence and multiple cloning site, that is MCS, within the LACZ. This helps in the use of broad spectrum restriction endonucleases and permits rapid visual detection of an insert. PUC18 and PUC19 vectors are identical apart from the fact that the MCS is arranged in just opposite orientation. PUC vectors consist of following elements: PMB1 rep replicon region derived from plasmid PBR 322 with single point mutation to increase the copy number. BLA gene encodes beta-lactamase which provides ampicillin resistance, which is derived from plasmid PBR 322. In this site is different from plasmid BR 322 by 2-point mutations. Key steps for cloning in plasmid vectors. First need is the foreign DNA or gene of interest. Then a cloning vector. The gene of interest can be ligated in cloning vector when a cloning vector and the gene of interest is digested with the same restriction enzyme. When both are cutted with restriction enzyme, the DNA ligases both ends including foreign DNA and cloning vector, and now it is called as recombinant plasmid vector, which is transformed into a host cell which are compatible for that cloning vector. When a recombinant vector is introduced into the host cell, it starts replicating and our gene of interest is amplified. Bacteriophage lambda and derived vectors. Bacteriophage lambda and derived vectors are widely used in recent era. These bacteriophages have capability to package into the phage capsid, and these have ability to get absorbed on the host cell. Bacteriophage lambda, a temperate bacteriophage of a family Styloviridae. Its host is E. coli K12. Bacteriophage lambda is the most studied bacteriophage with a fully sequenced linear double-stranded DNA genome of 49 kb. Bacteriophage lambda have 50 genes and 12 base pair complementary overhangs at the both ends known as cos site. Only half number of genes are essential it to infect and replicate and package its DNA into the viral capsid. The infection of bacteriophage lambda starts with absorption of bacteriophage DNA by the bacterium. After entering into the host cell, the linear double-stranded DNA molecule circularizes through the cos site at its end to form a circular DNA-like plasmid. The virion has an isohedral head of 55 nanometer diameter and a non-contractile tail approx 150 to 10 nanometer. It can be divided in three parts: left arm, right arm, and a central region. Head and tail proteins are coded by left arm. Right arm is responsible for DNA synthesis, regulation, and host lysis, and the central arm is responsible for integration and excision regulation. A bacteriophage lambda just adheres to the cell membrane of its host and it injects the DNA into the bacterium. After entering in the host, the linear double-stranded DNA molecule gets cyclized and through cos site at the ends to form a circular DNA-like plasmid. After circularization, it opts one of the two life cycles present in it. First is the isogeny cycle. It gets integrated into the bacterial genome and replicates and transferred along with bacterial genome. Second is lytic cycle. In lytic cycle, it replicates using host cell machinery and continue up to the death of the bacterium due to overproduction of virion particles. Bacteriophage lambda genome organization in capsid. In capsid, bacteriophage lambda remains in linear form of double-stranded DNA. Lambda DNA exists in a linear form in the bacteriophage and a circular form upon entering bacterium. The switch from linear to circular form occurs through complementation of the overhanging DNA ends at the cos site. Many of the genes required for the integration of lambda into the host chromosome or for a new phage replication and assembly are grouped together at lambda chromosome. It is a circular form of lambda in host cell. After entering the host cell, it gets circularized for the lambda start replication in rolling ball manner. Here in figure, the cos site can be shown with its complementary sequence. Bacteriophage lambda insertional vectors. There are two types of lambda bacteriophage vectors are found on the basis of accommodating foreign DNA. First is insertional vector. In insertional vectors, these are simplest lambda vectors similar to concept of plasmid vectors. To prepare an artificial insert lambda vector, the restriction site present for any restriction enzyme in the lambda vector is minimized to 1. To achieve it, restriction sites are deleted or a phase is searched for mutation in the restriction site. The packaging limit for any insertion lambda vector is between 37 kb to 51 kb, accommodating maximum of 14 kb and minimum of 4.3 kb foreign DNA. Lambda-based replacement vectors. The full-length lambda molecule have two identical restriction sites flanked by a stuffer fragment. The stuffer fragment is replaced by a foreign DNA during restriction cloning. The vector without the foreign insert cannot be packaged due to the size limitation. It means that a small size of vector cannot be packaged inside the viron. Insert size range between 10 to 23 kilobase. Examples are lambda EMBL3, lambda EMBL4 and lambda dash etc. Here in the figure you can see the stuffer segment of 14 kb that can be replaced by a foreign DNA of almost same size. The PMBL4 replacement vectors have capability to carry up to 8 to 24 kb insert size. The bacteriophage lambda cloning vector has a middle segment responsible for insertion excision that is called as IE region, and this region can be replaced by a foreign DNA with the help of two BAM-H1 sites present on the either side of IE region. Hence, in a cloning strategy described in figure, foreign DNA can be put into the vector and then allowed to infect the bacteria. In the presence of IE region, phage will integrate into the bacterium chromosome and continues the lysogeny cycle. But when IE region is disrupted and replaced by the foreign DNA, it will continue the lytic cycle and forms plaque. Replication in bacteriophage. In lambda DNA, rolling cycle mechanism is used before lytic cycle, and the cos site help in recognition by RE, that is recognition elements. It results into concatameric molecule composed of several linearly arranged recombinants having several cos sites. Replication in bacteriophage. Lysogeny pathway. The phage DNA become integrated into the bacterial genome via homogeneous recombination between ATTP and the bacterial genome ATTB site and is replicated along with the bacterial DNA. The prophage DNA remains integrated until it is induced a lytic pathway. In lytic pathway, large-scale production of bacteriophage particles, proteins and DNA occurs that eventually leads to lysis of the cell. Here in figure, you can see how two different mechanisms of lysogeny and lytic cycle pathway are happen. In one pathway, the cell goes lysis, that is called lytic pathway, and in other lysogeny pathway, the lambda genome get integrated in the bacterial genome and remains until the lytic cycle induced. In vivo packaging of a lambda DNA. In vivo packaging, lambda DNA involves first making pre-heads, structures composed of major capsid protein encoded by gene E. A unit length of lambda DNA is then inserted into the pre-head, with the unit length being prepared by cleavage of concatameric lambda genome at neighboring cos site. A minor capsid protein D is then inserted in the pre-head to complete the head maturation, and the products of other genes serve as assembly proteins, ensuring joining of the capsid into completed tail to complete head. Here in figure, you can see that genomic DNA get inserted in lambda. In vitro packaging of lambda with the help of a helper phage. The in vitro packaging of lambda occurs by utilizing two E. coli strains that bear lambda lysogenes containing different defects in the packaging pathways. A defect in producing protein E due to mutation introduced into gene E prevents pre-heads being formed in strain BHB2688, a helper phase. A mutation in gene D prevents the maturation of pre-heads with enclosed DNA. It is into the complete head in the strain BHB2690. This is also a helper phase. The components of the BHB2688 and BHB2690 mixed lysate, however, complement with each other deficiencies and provide all the products of the correct packaging. Consequently, a recombinant lambda genome can be constructed in vitro and packaged into mature lambda phage particles before being propagated and replicated in the E. coli cells. Phage vector is used to carry and replicate foreign DNA inside the bacterial host cell. The phage DNA insert into host chromosome by recombination. Phage lambda had short region of single-stranded DNA with complementary base sequence called as cohesive cos site. Base pairing between complementary cos site allows the linear genome to form a circular within the host bacterium. Circularized viral genome can be integrated into bacterial genome by homologous recombination between ATTP site of viral genome and ATTB site of bacterial genome. Cosmids. Cosmids are simple hybrid cloning vectors that resembles plasmids. Cosmid offers additional advantage over bacteriophage lambda-based cloning vectors as they have bacterial origin of replication. They are chimeric cloning vectors and consist of region from bacterial plasmid and bacteriophage lambda. They contain a flanking cos site and hence after their delivery into the host cell it adopts a circular form. As cosmids contain bacterial origin of replication, it can be maintained in bacteria as such. In addition, it has antibiotic resistance gene like tetracycline resistant gene and allows the selection of transformed host cells. The cloning strategy follows the similar mechanism as discussed before for bacteriophage lambda-based vectors and its outline. The example of cosmid vector is PJB8. Here in figure, you can see the orientation of different regions in a PJB8 cloning vector. As plasmids, cosmids contain an origin of replication and a selectable marker. Cosmids also possess a unique restriction enzyme recognition site into which DNA fragment can be ligated. After the packaging reaction has occurred, the newly formed lambda particles are used to infect E. coli cells. The DNA is injected into the bacterium like normal DNA, lambda DNA and circularized through complementation of the cos ends. The selection of transformants is made on the basis of antibiotic resistance and bacterial colonies rather than plaques will form that contain the recombinant cosmids. Since lambda phage particles can accept between 37 to 51 kilobase of DNA and most cosmids are about 5 kilobase in size between 32 to 47 kilobases of DNA can be cloned into these vectors. This represents considerably more than could be cloned in lambda vector itself. From this module, we learned about the various key steps for cloning in plasmid vectors, bacteriophage lambda, and derived vectors. insertional vectors, replacement vectors, lysogenic pathway, and lytic pathway. We also studied the in vitro packaging of lambda DNA with the use of helper phage and about various bacteriophage lambda derived cloning vectors. Thank you.